Welcome to today's lesson on the organization of the photosynthetic apparatus, a key section in understanding how plants convert light into life. In photosynthetic eukaryotes like plants and algae, chloroplasts are the specialized organelles where photosynthesis takes place. These chloroplasts house an intricate internal membrane system called the thylakoids. This is where all the light reactions of photosynthesis occur, thanks to the presence of chlorophyll and other light-absorbing pigments. Surrounding these thylakoids is the stroma, a gel-like matrix where carbon fixation reactions take place, such as the Kelvin cycle. The thylakoids organize themselves into two main structures, grana lamellae, which are stacked, and stroma lamellae, which are unstacked. This architecture creates distinct zones for different parts of the photosynthetic process. A chloroplast is enveloped by two membranes forming a lipid bilayer, collectively called the envelope. These membranes regulate the import and export of substances necessary for photosynthesis. Now let's take a quick evolutionary detour. Chloroplasts are semi-autonomous. That means they carry their own DNA and ribosomes and can manufacture many of their own proteins. But others still come from the nucleus. This supports the widely accepted theory that chloroplasts originated from cyanobacteria through a process called endosymbiosis. The thylakoid membranes are embedded with critical proteins, reaction centers, antenna complexes, and electron transport systems. These integral membrane proteins are carefully arranged to extend into the stroma on one side and the thylakoid lumen on the other. This unique orientation is essential for efficient energy and electron transfer. Let's talk about spatial organization. Photosystem II or PS2 is primarily located in the stacked grana, while Photosystem I or PS1 and ATP synthase are in the unstacked stroma lamellae. In contrast, cytochrome B6F complexes are evenly distributed across both areas. This lateral arrangement of protein complexes means that electrons and protons generated by PS2 need to diffuse across some distance to PS1 and ATP synthase. This distribution supports a shared pool of electron carriers, primarily plastoquinone, and not a one-to-one pairing between photosystems. In fact, in most plants, PS2 tends to outnumber PS1 with a typical ratio of 1.5 to 1. Altogether, this highly structured and spatially separated apparatus ensures that light energy is captured, transferred, and converted into chemical energy with maximum efficiency. And that's the beauty of the chloroplast, a marvel of biological architecture. If you found this explanation helpful, don't forget to hit the like button, subscribe for more plant science content, and tap the bell icon so you never miss a video. Drop your questions in the comments. We would love to hear from you. See you in the next one.